So thank you for the organizers to give me the opportunity to talk. Let me just okay. So I'm going to talk to you today about the efforts that we're actually conducting in my lab, but also really importantly, really in collaboration with many other people here at UC Davis, but then also in specifically Berkeley. So you have heard about the IGI, and those are basically the people I'm working with. I would also like to really start out with acknowledging funding agencies. And really here, importantly, all the funding we have been receiving specifically from the TED Audacious project really have allowed us to really assemble a large team. We have about 40 different people, 10 PIs, 15 PIs, really experts in many different fields, really allow us to address really substantial questions we need to address in order to really mitigate methane from ruminants. So I'm going to really talk to you today mostly about what I love the most, and those are microbes. And all of you know really that microbes are really essential in many different environments. And you can, for example, look at health-related issues. So if you think about yourself as a human being, I have to disappoint you, you are actually more microbial than human. The other aspect, if you think about health issues, if you have a microbiome in your body or any animal, if you kind of disturb that and the microbiome is not happy, you are actually also not happy. So about 30 diseases we can already directly link to this dysfunction of microbiomes. In agriculture, we also know that microbiomes are really important. So if you think, for example, in microbes that live in the soil, if you take them out of the soil, plants are not really productive anymore. When you put them back in, plants become really almost twice as productive as you would grow them without the microbes. Other really important aspect in agriculture is also the fact that microbes are the only organisms that actually can fix nitrogen, and with that you can actually make that available to plants and animals. So again, microbes really important. And then if you think about it in environmental space, so if you think of that you're breathing oxygen that would come from plants, yeah, that's true, but actually 30% of all the oxygen that you're actually breathing comes from microbes that are living in the ocean. So again, microbes really essential. And then not last but not least, if you think about, if you basically go to the bathroom, actually, and that is all cleaned up by microbes afterwards. So microbes really important in any aspects of life. Now in the past, what microbiologists like me and other people have been doing, we actually have to go in the field and we have to collect samples from the environments we love. So that could be somewhere in a cave, or you can go in the barn and reach into the animal, and get your samples, and then you need to go in the laboratory and put it in a plate, and you need to basically hope that your microbe you are interested in is actually capable of growing in would basically need to survive. Now, that's not always the case. Now, it's really still good that you actually can go in the environment and collect these samples and study them in the laboratory, but there are several limitations to that, and one of them is, for example, that you can only ask very limited questions. So how fast does it grow? But also what kind of products does it make? And that ability to ask that question and answer that question is really important, because that has really resulted in a lot of discoveries that have resulted in a lot of the biotechnological applications. Now, about 20, 30 years ago, when it became more common that you could actually do DNA sequencing, you were actually able to extract DNA from a microbe that you were growing in the laboratory, and you can actually generate that genome. So you get the entire genome and you get a blueprint, and that's really important. It's not really what this organism is doing, but it allows you to give an idea what that microbe might be doing under certain circumstances. But you can also look for specific genes that you might be interested in. You can take these genes and put them into yeast, for example, and get a lot of protein, and you can actually, again, put that into application. So that's always where the money is, right? Applications, so you want to actually be able to do that. Now, fortunately, about 10 years ago, Jill Benfield from Berkeley started out this revolution where you actually what we call metagenomics. And that is actually what we have heard about today before. What you basically do, just imagine you go into any environment, you grab a sample of what you're interested in, and I simplify it here, but you basically extract DNA from that sample, and all of that DNA is basically from microbes. You put that the microbial DNA on a sequencer, and that sequencer spits out some sequences, and you put it onto your computer, that does some magic, and then you get all these genomes back. Now, importantly is really that these genomes are from uncultivated microbes. Well, maybe 1 to 5% of all microbes that you have in the environment in your laboratory, you're missing out the majority of these microbes that are actually present in any given environment. And that means you cannot study and you cannot understand what's really going on in the environment. And it's kind of depicted here a little bit. I don't have a laser pointer. But on the right, the tree basically, when you have these genomes, you put these genomes together and you can figure out how are they related to each other. And you also see, for those people in the front, they can actually see these red dots. These red dots actually indicate, from that study that was about 10 years ago, it indicates genomes that have been assembled that have no representative isolate in culture. I can tell you with the data that we're generating now, the trees are much bigger and we see even more of these red dots appearing. So there's a lot of stuff that we just don't even know yet. Now, if you think about microbes, no microbe really lives alone. They're really, they hang out in groups just like any other animal. They are depending on each other. They basically communicate with each other. And what's really important is that these microbes really function as a group. And the function of that group is basically then also dictating the function of that environment that they're living in. So that can be soil, that can be your body, but that obviously is also the groups of microbes that are basically in the rumen. And so only if you understand them, how they interact with each other, only then you can actually successfully direct rumen function into the way you want to have it. I think I just put that on here, but I think most of you are familiar with that. But if you think of the rumen, of the cow, as a bioreactor. So I'm not an animal scientist, I'm a microbiologist, have been doing my PhD in biotechnology. For me, the rumen is really, or the cow is really a bioreactor that is really exciting because I can actually manipulate it in a way I really want to have it go. And I can test different things in the rumen that would not be, I couldn't do in any other environment. It's still relatively complex, but compared to other environments, it's extremely, I can extremely dictate where it should go. Now, if you think about the rumen, basically as your bioreactor, you put in some plant material and your microbiome, so basically they break it down into hydrogen, CO2, volatile fatty acids, and methane. So unfortunately, we know all that the methane is being released, and that's a huge problem, so we want to change that. But there's also good news, because we basically, some of the intermediates are VFAs that are actually utilized by the animals. And what we want to do is basically see how we can take hydrogen and the carbon, and instead of having it go into methane, we want to make sure it's actually going into these volatile fatty acids, so that the animals can actually take these volatile fatty acids and be more productive. And it goes back to what we have been hearing before. It's not only about reducing methane, but it's also about increasing feed efficiency, and I believe it all goes together thanks to the microbes, basically, how we can connect. Now, there are actually tools out there that we actually can utilize to manipulate microbiomes, complex microbiomes, and we all have used that, I would assume. So one example is our antibiotics. We take them when we are sick. They are, we can use them, but there are disadvantages. So they're not really specific. They're mostly targeting specific groups of organisms, depending if your antibiotic is basically based on a cellular function or cellular property. The other problem we have with antibiotics, as you all know, if we basically overuse it, we can see resistance occurring. We also can't really predict reliably what the outcome on the microbiome or on the animal is. So many different issues here. But really, what the last point is extremely important. It's not permanent. And so Paul just talked about this. You might have the fact that you have to basically re-administer vaccine. So it's just a problem if you have to do this again. Chemical inhibitors, they have advantages, but they also have some issues. They're effective in stopping certain mechanisms. So 3-NOP is a really great example, really, really highly efficient. It really stops methanogenesis there. But potential issues are that there's a loss of potential of nutrients. It's really extremely hard to scale. There might be some environmental impacts that we don't know of. So again, some trade-offs. And last but not least, again, it's not permanent. So you have to apply it over and over again. Now, if we are in a if you're in an industrial setting, that's fine. We have access to the animals. But if you're in a grazing system or low or medium income countries, that is certainly not a solution. So what we're going to do about this, and again, this is all kind of a team effort by the people at Berkeley and here at UC Davis. We want to basically come with a solution that is specific. So we want to be able to specifically target microbes, certain genes, certain pathways. So we want to be very clear about that. We want to have it very predictable. We want to have it permanent, and it needs to be scalable. And the way we're going to do this is basically taking advantage of some of the different rumen systems we have available to us. So we have animals, we have bioreactors in my lab. And so we basically will go and collect different rumen microbiomes from different animals in different stages. We'll take them in the lab, we'll actually manipulate that, and we can, all over that way, we basically collect not only DNA, but also RNA and in some cases proteins. So really the important point is here that we also want to look really at the phenotype. So we want to look at what genes are actually activated under these particular conditions. Then we do a little bit of modeling. It sounds so easy, but it will be harder than it is, and than it sounds. But then we come up with strategies that we basically will allow us to target these particular genes, particular microbes, and then we'll test it in the lab. And it's really important because we want to make sure everything is kind of exactly going the way we want it. Remember, I said things need to be predictable. And once we have that figured out, then we actually have the ability to actually apply it to the animals and test if we actually hopefully then see the effect in the animal itself. So there are two technologies that are really essential to this part. We talked about metagenomics before. I just want to reiterate that because it's really important. So again, here is the idea that you actually can access all the microbes in the environment, not only what you can culture in the laboratory. The other aspect is really we can at least predict what that microbe might be doing, and then if we actually add a different layer of metatranscriptomics, so we look at the gene expression profile of these microbes on top of it, we actually can also determine exactly which of these genes are activated. The other aspect then that we utilize is this CRISPR technology. I'm sure you guys have heard all about the CRISPR hype, but the important point is that this has been established by Jennifer Doudna, who is part of our team, and she received the Nobel Prize for that 2 years ago, I believe. But the important thing is here that we have shown that we can actually do this in complex communities. So the idea is you basically look at the genome, and those in the past have been genomes that were human cells. Also want to do it now in microbes we haven't isolated before using that metagenomic technology. So we know what the genome looks like, we can identify the targets, and we can go after it. And it's pretty much like it's depicted here. You basically find your target, you cut it open with some enzymes, and then you basically can insert or remove additional genes or pathways, and then you end up with a product with your microbe that is basically either having a different phenotype or it has a different phenotype, but it might have additional capability or it might have lost the capability. So really essential. And the cool thing again is you can actually apply it into mixed cultures, into real environments. Now, I just have one slide, but what I wanted to show you is here basically this is just an example of the insights we have been getting from utilizing some of these metagenomics approaches, and this is basically just focusing on the rumen microbiome. But really important, we started out doing this at the same time when Jill was basically establishing that metagenomics technology. The team I was working with and in Berkeley, we actually had the first rumen metagenome, so we assembled 25,000 carbohydrate degrading genes we put out 15 genomes of uncultured bacteria, and that's pretty much a joke these days, but at that time it actually was pretty substantial. And the last one I want just to point out is basically, if you look at the very lowest one, this is basically where we use a combination of metagenomics, metatranscriptomics, and metaproteomics, where we actually look at the archaea, bacteria, but also at the fungi that are in the rumen. And we could actually determine that specifically bacteria and fungi, who are extremely important biomass degradation, that they actually produce at any given time to really facilitate the biomass degradation in the rumen. So now getting a little bit more to the science and more related to what's going on now in the lab. So this is one of these bioreactors that we have established in the lab, and we'll get 2 more of these actually in the laboratory. And I just put up here the seaweed. So seaweed, as you all know, has been really shown to be very efficient. It's basically like nothing else but like a feed supplement. And what we used that bioreactor for is basically we wanted to see if we can actually take red seaweed and see enteric methane being reduced in the laboratory in these bioreactors. And then Brianna Rook, who was actually working on her Matthias, and she basically took these bioreactors. In half of the bioreactors, she put rumen fluid with the seaweed, and in the other half, she actually had rumen fluid without the seaweed. And in red, you can actually see, or you can't, but in the gray, you actually see the amount of methane is actually being produced in these bioreactors. And in the red, you basically, what you would see if there would basically be any methane, you would see a higher increase. So basically, Brianna could show that there was a reduction of 90% of methane in vitro. So in vitro is nice, but you also want to see really what's going on in the real animal. And so Brianna and Matthias at the same time actually performed an animal trial with the same seaweed, and what they actually found there, that methane was actually reduced by about 70%. Feed efficiency increased. Again, really that's great to see that both basically go together, and importantly also milk quality remained pretty much the same. Now, Brianna was smart enough to actually sample some DNA and RNA at the same time from these animals, and we generated some sequences. And now Spencer, who is actually sitting here in front, who is from the IGI, and one of his postdocs, and I have been looking at the data that was generated for us. So this is a phylogenetic tree. This looks a little bit different because we have more data, but the colored wedges are basically different families. And what you can see is basically each of the tip of these different branches is one of the genomes that we have sampled from our metagenomic data. And then you actually can also see in red, if you're sitting in the front, there are little red dots at the very tip of these branches. Those are actually genomes for which we also have transcriptome data, so where we actually see that they have expressed genes in our data set. Now in the outer ring, you actually see, in the inner 2 rings, you see control versus treatment, so animals with and without seaweed. You can't see the details, but you see differences basically where some of these genes or genomes are more active in the presence or absence of seaweed. On the red side, we are looking actually at methanogens specifically, so we can actually really look into these particular families, functional families of microbes. It's even getting better because we actually can now actually look also really at, more specifically, at bacteria, for example. So on the upper part, the bars where you can actually, you see changes, and the only thing you need to see that there are changes. On the left, I believe, is control, and the right is treatment. So you can see basically that it's not only methanogens that are actually affected by seaweed supplementation, but also bacteria. And again, this is actually transcriptome data. So these are expressed genes. So it's not anymore potentially what's going on. This is actually really what's happening. Now in the middle, what do you see? Message here is really, there are a couple of 3-letter words that you're seeing. And if they are in red, that means basically they are no longer expressed in the presence of seaweed. So we actually can determine really which of these pathways are shut down under certain circumstances. And again, I think it's getting even better because you actually now can look at particular genes. And it's difficult without. So let me see. Pay attention to where I'm pointing, trying to point. Hydrogenases, so the little boxes with the hydrogenases on them, with the asterisk on top, these are basically two hydrogenases. One is upregulated, highly upregulated, and the other one is downregulated. And if you look at the bar on the x-axis, basically those are hydrogenases that are involved in hydrogen consumption. And so when we looked at the one that the first box on the left that has a star, we looked at it that is upregulated. And it's really exciting because on the right panel, what you see that all these hydrogenase transcripts, so again, expressed genes, are coming from one genome. It has a funny name because it's one of these uncultured microbes. But when we look at that genome and we can assemble the entire genome and we can actually kind of figure out what's going on here. And again, I need to go and point that out, but what I want to explain to you, so there's this green box down on the left side. That is basically the depiction was upregulated. And when you look at that figure, what you can see, it's basically utilizing the hydrogen. And it's utilizing the hydrogen to facilitate fumarate fermentation. What that also means is basically, and at the same time you generate protons. These protons can be imported into the cell to generate ATP. And what that means is this organism has more ATP, has more energy, can grow better. And the important point is this is under supplementation of seaweed. So we actually know now that this might be a way where we can actually, where could we utilize our additional hydrogen for? Where can that go when basically there are no more cyanogens basically growing? So with that, just 3 more slides, because seaweed was a great story. We have been funded by CDFA. We are looking at local agricultural waste products to see if we find something similar, something that's of similar success than a seaweed. So hopefully we'll find some byproducts here. So Rich and Elizabeth had a poster early on. I think, unfortunately, there are no posters tomorrow, but if they're in the audience, you might still be able to talk to them. And Pedro just started his postdoc with me 2 months ago. He's actually looking at methanogens, and he has his first pretty promising results. It looks like he has already some novel methanogens in culture. And then we have Cass as some first fungi from the rumen. And so with that, I want to really acknowledge my team who has been doing the work, and then I really would like to thank you for your attendance and attention, and I'm happy to take any questions.